Hello everyone, I'm glad to see you on the course Biotechnology with Selection Basis. Today we have the theme of video lecture Biotechnology as a branch of science, the history of formation and development. Today we have the following goals. Learn about biotechnology as a science and industry and explore the following topics. First, directions of biotechnology. Second, sciences with which biotechnology is associated. Third, historical background, and last, fourth, areas of the use of biotechnology in general. Okay, let's start. Biotechnology is a branch of production based on the use of biological objects and systems. This concept includes a complex of production processes based on the use of biocatalysts, microorganisms, and various biological systems, such as cultures of plant and animal cells and tissues, immunocorrection, and manipulation of germ cells. Biotechnology arose from the use of microorganisms and biosystems with programmed properties based on the achievements of genetic engineering and engineering enzymology. The main directions of biotechnology include the following. First, genetic engineering. It is a field of molecular genetics that develops methods for constructing new genetic programs. Second, cell engineering. Generation of cells of a new type, hybridoma technology, construction of genetically new objects by cell hybridization, and introduction of foreign genetic material. Third, embryogenetic engineering. This is an active restriction of the animal genome by interfering with their development at the early stages of ontogenesis. Fourth, traditional biotechnology. It is about the use of anaerobic processes for the production of wine, silage, fermentation, obtaining lactic acid products, alcohol, and more. And fifth, last, engineering enzymology. It is application of microbiological, physical, chemical methods for the production of enzymes, specific catalysis of a protein nature. More information about embryogenetic engineering. Genome rearrangements. It is the reconstruction of embryos by cloning, fusion, or injection foreign DNA into their nuclei. The main directions of embryogenetic engineering include the following: animal cloning, obtaining genetic kinemas, obtaining transgenic animals, and embryo transplantation. Biotechnology is closely related to many sciences, such as biological and bioorganic chemistry, molecular biology, genetics, microbiology, immunology, physiology of animals and humans, cytology, and more. Historical periods of biotechnology. The foundation of modern biotechnology is molecular biology, microbiology, genetics, biochemistry, biophysics, technology, instrumentation, and more. Over the past 40 and 50 years, there has been an upward development of these sciences, which has led to a revolution in the production of veterinary and medical biological products, the creation of transgenic plants and animals with given unique properties. Such studies are priority areas of scientific and technological progress, and in the 21st century will take a leading place among all sciences. Science was formed and involved with the formation and development of human society. Its emergence, formation, and development can be conditionally divided into four periods. The first period, empirical or prehistoric. From the Greek language, empiricals means experienced. This period is the longest, covering about 8,000 years, of which more than 6,000 years BS and about 2,000 years AD. The ancient peoples of the time intuitively used the techniques and methods of making bread, beer, vinegar, obtain ferment milk products, pickling cabbage, and siling, which we now refer to the category of biotechnology. The second period, etiological, from the Greek language atia means the reason. This period in the development of biotechnology covers the second half of the 19th century and the first third of the 20th century. From 1850 1956 to 1933. It is associated with the outstanding research of the great French scientist Louis Pasteur, the founder of scientific microbiology. Pasteur established the microbial nature of fermentation, provides the possibility of life in anoxic conditions. Experimentally referred, then existed notion of the spontaneous generation of living beings, created the scientific basis for vaccine prevention and vaccine therapy, proposed a pasteurization method as a sterilization method. In the same period, his outstanding students, collaborators, and colleagues were engaged in this science. For example, Duclos, Roux, Mechnikov, Koch, Lister, Kitazato, Ivanovsky, and others. Among the scientific achievements, it is worth noting the following. 1868. Mecher received nuclein DNA from leukocytes. 1902. Haberland showed the possibility of cultivating cells of various plant tissue in the simple nutrient solution. 1912. Neuberg discovered the mechanism of fermentation processes. 1913. Michaelis and Menton developed the kinetics of enzymatic reaction. The next period, the third biotechnological period, from 1933 to 1972. In 1933, Kluwer and Perkin published the work Methods for the Study of Metabolisms in Molds, in which they outlined the basic techniques as well as approaches to assessing the results obtained in deep cultivation of fungi. The introduction of large-scale, hermetically sealed equipment into biotechnology, which ensures the conduct of processes in sterile conditions, began. A particularly powerful impetus in the development of industrial biotechnological equipment was noted during the formation and development of antibiotic production during the Second World War, when there was an urgent need for antimicrobial drugs for the treatment of patients with infected wards. All progress in the field of biotechnological and technical disciplines achieved by that time was reflected in biotechnology. So the next dates. 1937, Krebs discovered the 3-carboxyl acid cycle, and in 1953, Crick and Watson deciphered the structure of DNA. It's a great discovery of the world. These facts became an incentive for the development of methods for large-scale cultivation of cells of various origin to obtain a variety of cell production and cells themselves for human needs, and above all, penicillin, streptomycin, tetracyclines, dextran, and number of amino acids, and many other substances. And last period, for biotechnology period, genetic engineering, from the Greek language. Genesis means origin or birth. This year in the USA, Berg in 1972 created the first recombination DNA molecule, thereby demonstrating the possibility of direct manipulations with the genetic material of bacteria, and more. And naturally, without the fundamental work of Crick and Watson in 1953 to establish the structure of DNA, decipher the genetic code, it was impossible to achieve modern results in the field of biotechnology. And elucidation of the mechanisms of function and replication of DNA, isolation and study of scientific enzymes led to the formation of a strictly scientific approach to the development of biotechnological processes based on genetic engineering manipulation, of course. And the most important achievements of biotechnology in the fourth period. First, development of intensive processes instead of extensive ones, based on directed fundamental research with superproducts of antibiotic, enzymes, amino acids, vitamins, and more. Second, creation of various products necessary for a person on the basis of genetic engineering technologies. Third, creation of unusual organisms that did not previously exist in nature, non-nodal plants carrying the gene of nitrogen bacteria, which are responsible for the ability to fix molecular nitrogen from the air, as a result of which there is no need to fertilize the soil with nitrogen-containing fertilizers. Glowing plants obtained in chimeras, sheep goats in the USA, ducks in Russia, potato hybrid of potato and tomato, animal cloning, and more. Fourth, development and implementation in practice of special equipment by technological schemes. 5. Automatic and computerization of biotechnological optimal production processes with the maximum use of cheap raw materials and minimum energy consumption. And last, the introduction of biotechnology in animal reproduction. Embryo transplantation from a donor to recipients allow you to get more than 100 calves from outstanding cows and speeds up the selection process 2 or 3 times. In general, the application of biotechnology in the national economy is very diverse. Areas of the use of biotechnology. The following are only the main areas of application, since methods are currently being developed to obtain more than 1,000 names of products by technological methods, and it is impossible to list the industries in which they are already used or can be used. For example, medicine, healthcare, pharmacology, obtaining chemicals, animal husbandry, crop production, fisheries, food industry, energy and mining, heavy industry, light industry, bioelectronics, astronautics, ecology, scientific research, and more. Okay, we have considered full material in the framework of this exam. Thanks for your attention and goodbye.